Okay, we'll get started. Good afternoon, everybody. I'm pleased to bring you back to the Connect Science seminar series and have to apologize. I've got dogs barking in the background here. But a few housekeeping notes, if hopefully you can hear me before we get started, is to please post any of your questions in the Q&A tab, not the chat tab, so that at the end of the lecture I can pose the questions, and anything that we don't have time to get to will get emailed to Dr. Wargo so she can connect with you after if that's something that you wish to do. And the talks are recorded, as you see at the website that you see on your screen here is where you can find the recording of the talks. So with that, it is a real honor and pleasure of mine to introduce Dr. Jennifer Wargo. She's coming to us from MD Anderson, where she's a professor both in the Department of Genomic Medicine and also surgical oncology. She also has an endowed professorship there as the Dr. R. Lee Clark professor. She's no stranger to Boston. She was here years ago at Harvard to get her master's in medical science, and then did her clinical internship and residency at Mass General. She then also did a research fellowship in surgical oncology at UCLA, and came back to Boston, and then was also did a clinical fellowship at the NCI. Her long list of accomplishments and accolades and awards and long list of publications really speak to how well regarded she is in the field and the wider research and clinical research communities. It really speaks to her openness and collegiality, which she's also known for. So it's clear she's made seminal contributions to the field of immuno-oncology, and particularly looking at response to therapy, particularly immune checkpoint blockade in melanoma and other solid tumors, and has really focused efforts on understanding how immune fitness can impact response to therapy. And one really fascinating line of investigation is how the microbiome impacts immune fitness, and therapeutic response. And she's really translated her work back and forth between the clinic and the bench. And so we're really, really fortunate to have her here today. And with that, I will stop and let Dr. Wargo take the screen. Welcome.
Thank you. Thank you, Sandra. I appreciate it. All right. Well, it's my great honor to be back in Boston, if you will. And today I wanted to talk to you a little bit about how we can better understand responses to cancer therapy, really through tissue-based analysis, but also through studies on the microbiome. And I'll give you some important information on how the tissue is the issue, the scoop is in the poop, and you are what you eat. And so I do have a few disclosures. And so it's great to be back in Boston with friends at Dana Farber and the Harvard Cancer Center. As Sandy said, I spent almost 20 years in Boston, and I actually got married on a beach in P-Town to my partner, Haven. And in addition to loving Boston from a personal standpoint, also have a tremendous number of collaborators in Boston, and hopefully that number will grow even more after today's talk. And that's including phenomenal investigators like Steve Hoody, Arlene Sharp, Sandy, Tony Latai, Curtis Huttenhauer, Wendy Garrett, and the list goes on and on. Beth Mittendorf, who recently moved from MD Anderson. And as Dr. McAllister highlighted, I did my surgical residency at MGH, and then did a research fellowship in cancer immunotherapy with Jim Economo and Tony Rebus. I then did a surgery branch fellowship with Steve Rosenberg down at the NIH, was on faculty at MGH for about 5 or 6 years, and then moved down to MD Anderson now about 8 years ago. And so what I wanted to do in the next 40 minutes or so is really highlight how we can better understand responses to cancer therapy. I'll first go through a background. I'll talk a little bit about lessons learned from translational research on tumor tissue and the microbiome, highlighting that the tissue is the issue, but the scoop is in the poop. And finally, I'll talk about the role of diet and other factors in therapy response, and talk about you are what you eat, and give you a little lunchtime advice, if you will. And so we've certainly made major advances in cancer treatment with the use of immunotherapy, with an overall decline in cancer mortality. And I show some of the recent approvals for immune checkpoint blockade, as well as data regarding mortality rates for cancer. But what I would argue is that there's still a critical need to improve responses as well as to limit toxicity to cancer therapy in patients with established cancer. There's also a real opportunity to prevent cancer altogether. And so I highlight this with a case. And so this is a patient I treated about a year or two ago. And this is a 44-year-old male who came in to see me, had a history of multinodular goiter. He actually developed locoregional metastatic melanoma and came in to see me here. And you can see he had bulky lymphadenopathy in the right axilla. He was treated with neoadjuvant combined immune checkpoint blockade targeting CTLA-4 and PD-1, actually had a complete resolution of any metastatic disease by imaging. But we know that in addition to having these phenomenal responses, we know that these treatments can actually be associated with a high rate of immune-related adverse events. And this patient had just that. So during treatment, he actually developed thyrotoxicosis, lost 50 pounds, was profoundly hypothyroid afterwards, had enlargement of his thyroid, and actually had tracheal compression with the delay in surgery. However, when we took him to the operating room, eventually, a couple of weeks later, I took out all his lymph nodes and he had no viable tumor, and he's alive and well and is on lifelong treatment with thyroid replacement hormone, but a small price to pay, if you will. And so I think this case highlights, the tremendous advances, but also some room to grow there, if you will. And we know that a powerful way to better understand responses as well as toxicity to cancer treatment is through this process called reverse translation, where we can take findings from our patients at the bedside back to the bench and then back again. And we've done this in a manner in which we collect longitudinal tissue, blood, and other samples in the context of treatment, do deep profiling of these tissues and blood, and then go back to mouse models to perform mechanistic studies and therapeutic optimization, and have been able to really turn this around real time and to bring advances back to our patients. And I'll show you some examples of that. And so what we know by doing these types of studies is that responses as well as toxicity to immunotherapy and other treatments are dependent on a number of factors that shape tumor growth and immunity. And so we know certainly with the advent of next-gen sequencing that the tumor genome and epigenome are critically important. We also know that the tumor microenvironment plays a major role. Systemic immunity, of course, and particularly with regard to cancer immunotherapy, but now we know that environmental factors such as the microbiome can actually have a tremendous impact. And so first, I'll take you through some stories that highlight the importance of sampling in the context of therapy and as well as some microbiome studies. And this is the part of the tissue is the issue, but the scoop is in the poop. And so tissue-based analyses have helped us understand how oncogenic mutations can impact anti-tumor immunity. And this is work that I started while I was on faculty at MGH, where we basically, at the time, immunotherapy wasn't working, if you will, but targeted therapy was. And so we wanted to better understand response and resistance to treatment with targeted therapy in patients with melanoma. And so we profiled tumors of patients. And what we found is that if you target oncogenic BRAF, you can actually improve anti-tumor immunity. But at the same time that you get an increase in melanoma antigen expression and an increase in T-cell infiltrate, you get upregulation of ligands for immune checkpoint inhibitors. And so we know that as soon as those T-cells are getting in, they're being shut down. And this provided the rationale for combining treatment with molecularly targeted therapy and immune checkpoint blockade in melanoma and other cancers. And this has now actually borne out real time. We know that by combining treatment with targeted therapy along with treatment with immune checkpoint blockade, you can actually really improve responses. And this has been shown in melanoma, as shown here in these publications in Nature Medicine. And interestingly, it's being studied in other cancer types. Most recently, KRAS. So these KRAS inhibitors can be combined with immune checkpoint blockade to improve therapeutic responses. In addition to this, we know that with regard to immune checkpoint blockade, we and others have certainly looked at tissue-based biomarkers of response and have found some novel ones, including B cells and tertiary lymphoid structures with the 3 papers that we co-published in Nature last year. And there's a real opportunity to target these therapeutically in order to improve response to treatment. And importantly, treatment with immunotherapy and other strategies are actually being used in patients with earlier stage disease. So it's not just being used in patients with advanced disease, but now in patients with earlier stage disease, we're using these, and tissue and blood-based analyses are really critical here. And I show data here suggesting that treatment with immune checkpoint blockade after surgery is associated with improvement over placebo. But we know that there's also a very strong rationale to use these in the neoadjuvant setting or the pre-surgical setting. And I show here an example of a patient who came in with bulky disease either in the groin or in the axilla. And we know that for patients with local regional metastatic melanoma, upfront surgery is currently the standard of care. But up to 70% of patients who were taken straight to surgery could actually relapse and die of disease, at least before the advent of treatment with adjuvant targeted therapy or immune checkpoint blockade. So there's a real clinical rationale to treat patients with these agents before surgical resection. In addition, there's a real strong preclinical rationale. And we know that from studies in mouse models that if mice are treated with neoadjuvant immune checkpoint blockade rather than adjuvant, they actually have much better survival. And there are tremendous opportunities to incorporate tissue, blood, and other analyses during these studies. And so we actually first studied the use of neoadjuvant targeted therapy versus standard of care upfront surgery in patients with high-risk resectable metastatic melanoma. What we did is we hypothesized early on that if patients came in and had bulky stage 3 melanoma, if you treated them before surgery, rather, that they would actually have better long-term outcomes compared to upfront surgery. And so we designed a trial to answer that question, where we took patients with bulky stage 3 melanoma who had a BRAF mutation and randomized them to either get standard of care upfront surgery, what they'd get if they walked into any surgeon's office in the country, versus treatment with neoadjuvant and adjuvant BRAF and MEK inhibitors. We treated them for 8 weeks, did surgical resection if they did not progress on treatment, and then treated them in the adjuvant setting as well. And I show you here that not only did we run this as a clinical trial, we also got longitudinal tumor and blood sampling and did deep molecular and immune profiling on these. And what we found when we ran this trial is that patients who were treated with neoadjuvant and adjuvant BRAF and MEK inhibitors actually had high response rates. 85% of them had a RECIST response. 58% of them had no viable tumor at time of surgery. And when we looked at the relapse-free survival in these patients, to no great surprise, we saw that if patients were treated with neoadjuvant and adjuvant BRAF and MEK inhibitors, they actually had better long-term outcomes compared to upfront surgery, those treated with upfront surgery. And if they achieved a path CR, virtually 100% of those patients actually derived therapeutic benefit. Whereas if they failed to achieve a path CR, very few of those patients did. And other groups have made similar observations, and this is data from Georgina Long, who also made similar observations with regard to treatment with neoadjuvant and adjuvant BRAF and MEK inhibitors. Now, in addition to treatment with neoadjuvant BRAF and MEK inhibitors, we next studied the use of neoadjuvant immune checkpoint blockade in patients with high-risk resectable metastatic melanoma. This time randomizing patients not to standard of care upfront surgery, but randomizing them to either treatment with PD-1 inhibitor alone versus combined treatment with CTLA-4 blockade as well as PD-1 blockade. And what we found when we did this study is that if we treated patients with combined immune checkpoint blockade targeting CTLA-4 and PD-1, they actually had a much better response rate, about 73%. RECIST response rate versus 25% with PD-1 monotherapy. And the path CR rate nearly doubled. It was 45% compared to 25%. But what the hitch was is that there was a lot of toxicity associated with this regimen. We used IPI 3 mg/kg and NIVO1, and 73% of the patients treated with combined immune checkpoint blockade had grade 3 or above adverse events, as opposed to only 8% with nivolumab monotherapy. And so lessons learned there. Now, importantly, we have been working with other groups to really optimize these neoadjuvant strategies and to help define the optimal regimens. And this includes with the group from Penn, who showed that with a single dose of a PD-1 inhibitor, pembrolizumab, they could actually see 25% of patients, up to 25 or 30% of patients that had a major pathologic response, and that was associated with long-term benefit. And then Christian Blank and the group at the Netherlands Cancer Institute in this OPUS and NEO study that was published in Lancet Oncology a year or two ago actually showed optimal dosing regimen with IPI at 1 mg per kg and Nivo at 3 mg per kg, where they could preserve the response rate and actually reduce the toxicity. And we are working with people worldwide to really translate this, including with the Melanoma Research Alliance, with the FDA, with industry, and with other investigators to build on this and to improve patient outcomes. And this includes the International Neoadjuvant Melanoma Consortia, which we formed a few years ago to help address these types of questions, have put out several white papers based on our insights, and also had a workshop with the MRA and with the FDA just about a year and a half ago, which has really helped to define really the use of these neoadjuvant strategies in patients with local regional metastatic melanoma. And I'll highlight a recent publication from the International Neoadjuvant Melanoma Consortia, two recent publications in Nature Medicine, one of which showed that pathologic complete response was associated with long-term benefit in patients, regardless of whether they were treated with either targeted therapy shown over here versus treatment with immune checkpoint blockade. But there were some notable differences. And if you look at this pooled analysis, what we see is that, yes, patients who achieve a pass CR on targeted therapy do much better than those who fail to achieve a pass CR. But it's really only those patients who have absolutely no viable tumor that derive the benefit. And even the patients who have a pass CR, some of those patients will relapse, and often, interestingly, within the brain. Now, contrast that with treatment with immune checkpoint blockade in the neoadjuvant setting, and you can see that, if patients have a path CR, they actually do quite well. And even if they don't have a pathologic complete response, they do really pretty well. And if you look at the plots below this with different degrees of a pathologic response, it's only the patients who have absolutely no response after neoadjuvant treatment that don't do well, even if you have some pathologic response to neoadjuvant immunotherapy, you can really derive long-term benefit. And Christian Blank and others have now gone on to look at tissue-based biomarkers, which could predict the responses. And sure enough, it's tumor mutational load as well as interferon signatures. And that's shown in this paper that was recently published here. Now, this does include efforts to use more personalized approaches after neoadjuvant immune checkpoint blockade. And this is a PRADO trial that's being run by Christian Blank and others at the Netherlands Cancer Institute, where they basically take patients with bulky stage 3 melanoma, treat them with neoadjuvant immune checkpoint blockade using that optimal regimen, just resect that index node rather than doing a complete lymph node dissection. And if patients have a pathologic or a near pathologic complete response, they actually don't treat them further. They do not do a completion lymph node dissection, and they just follow them up with ultrasound and CTs. However, if they have only a partial response, they go on to have a therapeutic lymph node dissection and then are followed. And if they fail to derive any benefit on pathologic analysis, they not only undergo the therapeutic lymph node dissection, but are also treated in the adjuvant setting with either targeted therapy or with immune checkpoint blockade. And interestingly, in this study, therapeutic lymph node dissection was actually omitted in about 60% of patients. And this is treatment with immune checkpoint blockade is actually in the neoadjuvant setting is being used in other cancer types with success. And I show a couple of studies here for patients with non-small cell lung cancer. This includes from MD Anderson Cancer Center as well as from Hopkins, where groups have showed that if patients with non-small cell lung cancer are treated in the neoadjuvant setting, actually many of those patients will derive long-term benefit. And in addition to that, others are looking at other tumor types, and certainly this is a trial being run by Chrissy Rowland and others looking at patients with either different forms of sarcoma. And you can see that patients will often have responses. These are the pathologic responses. And when treatment with immune checkpoint blockade was actually combined with radiation, nearly all of the patients had no viable tumor at time of surgery. And what does this mean for us long-term? Well, this is a presentation from a presentation that Lex Egermont gave a few weeks ago with me, and basically, showing that the number one neoadjuvant immunotherapy really landscape in the next 5 years is more cures and less surgery. So if you look treatment across multiple tumor types, melanoma, bladder cancer, MSI, colorectal cancer, and multiple other cancer types, certainly I think we'll be seeing more use of neoadjuvant immune checkpoint blockade and hopefully a lot less surgery. So that's really, the tissue is the issue part, but considering that humans and organisms represent a complex ecosystem, what's the role of microbes that coexist with us on cancer and therapy response? And this is the scoop is in the poop part, if you will. And so why should we study the microbiome? Well, an interesting little piece of trivia is that there are actually more genes in the human microbiome than there are stars in the galaxy, kind of a cool little piece of trivia. And disturbances in the gut microbiota and microbes in other niches are actually implicated in several different disease states. And that includes an inflammatory bowel disease, colon cancer, heart disease, but also now in autism and even in other forms of cancer. And I posed the question, could the microbiome actually become the newest frontier in oncology and even in medicine overall, with diagnostic and therapeutic strategies that target the microbiome? And so interestingly, we first studied the role of the microbiome in cancer, but we did it somewhat serendipitously. And so I'll take you through that story. And so again, when I started my faculty position at MGH, at the time, immunotherapy wasn't working, if you will, and targeted therapy was. And what we wanted to do was to use our tissue-based analyses to really try and understand stromal-mediated resistance in melanoma. And so we worked with a group from MIT and the Broad with Todd Golub and Ravid Straussman, who had developed this system to look at just that, really hypothesizing that components of the tumor microenvironment alongside tumor and immune cells could actually mediate therapeutic resistance. These tumors aren't just balls of cancer cells, but there's multiple other cell types that could be contributing. And so to help answer this question, Ravid and Todd developed this 3D culture system where they co-cultured cancer cell lines, in this case melanoma cell lines, along with stromal cell lines and along with drugs, in this case targeted therapy or BRAF agents. And what they did is they then looked to see if there were any stromal cell lines that could mediate therapeutic resistance. And they did find several cell lines that were able to confer therapeutic resistance, so seen over here, found that these were actually secreting hepatocyte growth factor. Turned to us, Keith Flaherty and I, and we looked at human tissues and found, lo and behold, if patients had HGF within their tumor microenvironment, they were actually resistant to treatment with BRAF inhibitors. And we published these findings in Nature almost a decade ago. Now, we next wanted to use the same model to study stromal-mediated resistance to other cancers, and here's where the twist came in. And so at the time when I was on faculty at MGH, treating patients with melanoma, I was also treating patients with pancreatic cancer. I always say I was doing wide excisions and Whipples, and wanted to understand stromal-mediated resistance to pancreatic cancer and other solid tumors. And so used, again, the same 3D culture system to study resistance to chemotherapy in colorectal cancer and pancreatic cancer, this time co-culturing the cancer cell lines, either pancreatic cancer or colorectal cancer, along with stromal cell lines and along with drugs, in this case chemo. And what we found when we did this is that there was actually this one stromal cell line, HDF, or human dermal fibroblast line, that was actually able to confer therapeutic resistance across multiple different colorectal and pancreatic cancer cell lines. And it was highly reproducible. We were super excited about this. Again, found it was a secreted factor, scoured human tissues for potential enzymes, which we thought it was, and totally came up dry. But then on routine testing of the cell line in the laboratory, we actually found that it was contaminated with mycoplasma, which at first was a bit devastating, but Ravid Straussman is brilliant. And he said, well, wait a second, what if this mycoplasma is actually contributing to the therapeutic resistance? And so to test that, he actually eradicated the mycoplasma from the cell line, no longer conferred rescue. He infected another cell line, conferred rescue, did whole genome shotgun sequencing of the HDF preconditioned media, found mycoplasma, and found that these bacteria were actually breaking down gemcitabine into its inactive form. And so as a surgeon scientist, I was able to lend a helping hand. Actually, at Mass General would do resections for pancreatic cancer, sterilely harvest tumor, run it back up to Jackson 9 and plate out these tumors onto agar plates. And what we found is that if we resected tumors of patients with pancreatic cancer, these were chock full of bacteria. Whereas if we did resections for non-cancer indications, such as chronic pancreatitis, these were devoid of bacteria. We then validated this using different sequencing as well as visualization approaches, worked with Curtis Huttenhauer and others to perform metagenomic sequencing and found that these were largely gamma proteobacteria, and then took it back to a preclinical model and showed that by co-targeting these intratumoral microbes using antibiotics along with treatment with chemo for the pancreatic cancer, we could actually cure all the mice of their tumors, and published these findings in Science a few years ago. And we know now that intratumoral microbes may also impact anti-tumor immunity, with some of these microbes negatively impacting it, while others can actually promoting it, potentially even by neoantigens. And so this is beautiful work I show here from George Miller, who showed that bacteria could actually be found in patient tumors and in genetically engineered mouse models of pancreatic cancer, and that these were associated with a more immunosuppressive tumor microenvironment. But additional work done by Florencia McAllister actually showed that in some cohorts of patients with pancreatic cancer, the presence of these microbes was actually associated with better outcomes. And she showed in 2 independent cohorts of patients with pancreatic cancer, one from Hopkins and one from MD Anderson, in long-term survivors, she actually found a more diverse tumor microbiome, which was associated with more CD8 positive T cells in the tumor microenvironment. And how might these microbes actually be impacting anti-tumor immunity? Well, actually, Yardenia Samuels just published a paper in Nature 2 weeks ago or so, showing that microbes could actually be identified in melanoma tumors, and that microbial neoantigens are actually being processed and presented by these tumor cells and recognized by T cells. And microbial signatures are actually now being identified across tumor types, and opportunities to target them to improve outcomes are really emerging. And could we even prevent cancer altogether? And so 2 recent publications, one by Ravid Straussman in Science in May of 2020, and one by Rob Knight in Nature about a year ago, showed, first, Ravid showed that intratumoral bacteria could actually be identified in all tumor types, though the composition differed depending on the histology. Rob Knight also mined TCGA data looking for microbial signatures and identified microbial signatures in tumors, pan-cancer, and even in the blood of patients with cancer. And I posed the question that these intratumoral microbes could serve as important biomarkers and maybe even as potential targets. Could we prevent cancer altogether by targeting these intratumoral microbes? And I think the answer could potentially be yes. And there's a precedent for this. Look at HPV. And we and others are now studying the role of bacteria and other microbes, including viruses, in carcinogenesis and in metastasis. And we were able to recruit a phenomenal PhD scientist, Gulnaz Murad, from Harvard. She's now with our group, and she's looking at the contribution of microbes within the blood, the tumor, and in the gut on metastasis in specific models. And she's actually working with the team here to mine data sets, and has actually found microbial reads not only in brain metastases, in primary tumor, in normal, and even in blood, and does see some differential signatures. And she'll be presenting this at AACR in the form of a poster. However, we know now that in addition to microbes within the tumor, we know that microbes within the gut can profoundly impact our physiology. And though our immune system actually originally evolved to fight invading pathogens, there's a complex and dynamic interplay between microbes and immune cells, and particularly within the gut. If you think about it, all along this long alimentary tract, on one side you have this carpet of microbes, and just on the other side you have this matrix of infiltrating immune cells, and there's this constant interaction and potential education of these immune cells by these microbes. And so landmark studies were performed several years ago that demonstrated that gut microbes could actually influence responses to cancer immunotherapy. Some of the earliest work was presented out of Memorial Sloan Kettering, where investigators like Eric Paymer and Ying Tower showed that in patients with AML going on to stem cell transplant, if they had a more diverse gut microbiome, which is generally associated with better gut health, if you will, they actually had better long-term survival in the setting of stem cell transplant for AML. So diversity does seem to matter. In addition to this, Laurent Zitvogel and Tom Gajewski in back-to-back papers in Science a few years ago showed that not only does diversity matter, that the composition of the gut microbiome seems to matter quite a bit. And in independent studies, they showed that in mouse models of melanoma and other cancers, the composition of gut microbes actually dictated whether or not mice responded to treatment with immune checkpoint blockade. And furthermore, that you could change the microbiome and make mice respond better. And so we saw this as a unique opportunity. I had seen the data presented at SITC and saw this as a unique opportunity to study this in patients. And so at MD Anderson, we opened a protocol where we enrolled patients on to study the gut and oral microbiome if they were going on to systemic therapy. And so in a year and a half, we collected samples from over 200 patients. We now have samples from over 500 or 600 patients with some follow-up data currently under review. And basically, I show the schema here where we took patients with metastatic melanoma coming in, going on to systemic therapy, collected an initial oral and gut microbiome sample, did a tumor biopsy when feasible, started therapy, restaged, and repeated the microbiome sampling. And so I show here the landscape of treatment, and in line with current strategies, most of the patients are treated with immune checkpoint blockade, and the vast majority were treated with anti-PD-1. So we focused on that cohort first. And so what we did in this cohort is we did microbiome sequencing as well as immune profiling, both at the level of the tumor as well as at the level of the blood. And what we found in these studies was that patients who responded to treatment with anti-PD-1 had a much more diverse gut microbiome, which was associated with better long-term outcomes, very reminiscent of what they had seen in stem cell transplant patients. And in addition to diversity, we also saw that there were compositional differences between patients who responded versus failed to respond, with responders to therapy having a higher abundance of specific taxa, such as Ruminococcus fecali bacteria and Clostridiales, whereas non-responders had a higher abundance of Bacteroideales. Now, importantly, these favorable signatures in the gut microbiome were associated with enhanced immune responses in the tumor microenvironment and even at the level of the blood. And so, again, we saw that responders to treatment had a higher frequency of CD8 positive T cells in their tumor microenvironment. No great surprise there. But when we compared the abundance of specific taxa in the gut compared to expression of cytolytic T cell markers in the tumor microenvironment, we found that if they had a favorable gut microbiome with a high abundance of Clostridiales, Ruminococcus, or fecali bacteria, they actually had a high expression of these cytolytic T cell markers. Whereas if they had a high expression of the bad bugs in their gut, Bacteroideales, they actually had a low expression of these cytolytic T cell markers in the tumor microenvironment. And we saw similar patterns in the blood. We then performed mechanistic studies in germ-free mouse models and essentially recapitulated this phenotype. What we did is we took fecal samples from patients who either responded or failed to respond and performed an FMT, or a fecal microbiota transplant, into germ-free mice, implanted tumors treated with checkpoint blockade, and showed that if mice received a fecal transplant from a non-responding patient, the tumors grew rapidly and failed to respond to treatment with immune checkpoint blockade. Whereas if they received a fecal transplant from a responding patient, the tumors grew slowly and responded brilliantly, rather, to treatment with immune checkpoint blockade. And we now have performed additional studies suggesting that this is mediated both at the level of the gut and the mesenteric lymph node, but also potentially by metabolites that are being produced by gut microbes. Now, in our cohort, we did identify a gut microbiome signature, if you will, with a high likelihood of response, a so-called type 1 signature, where patients with a type 1 signature had a high abundance of Ruminococcus fecali bacteria and other bugs. And if they had this type 1 signature, virtually 100% of those patients responded. Whereas if they had a type 2 or a less favorable signature, only half of those patients responded. And together, this suggests that the gut microbiome could actually be used as a biomarker of response to immune checkpoint blockade, with patients having a more favorable signature that are more likely to respond. And reassuringly, numerous studies have actually been done in human cohorts that support a link between the gut microbiome and response, as well as toxicity to cancer therapy. And I show a couple studies here. These were studies that were co-published by Tom Gajewski and Laurent Zidvogel in Science in 2018, which showed that additional patients with melanoma and also patients with non-small cell lung cancer or kidney cancer, that differential signatures in the gut microbiome were seen between responders and non-responders to therapy. And additional studies have been published on toxicity to therapy. Now, a big question that arises out of this data is, can we actually modulate the gut microbiome to enhance response to immunotherapy and or to abrogate toxicity? And I can tell you, you can guess my answer to this. It's totally yes, but we don't have the perfect ways to do that just yet. But there were two papers that were recently published in Science that really support the role of gut microbiome modulation using FMT or fecal transplant in improving responses to treatment in patients with melanoma. And so the first of these was published by Erez Baruch and others from the Sheba Institute, and showed that fecal microbiota transplant could actually promote response in immunotherapy refractory melanoma patients. And so what they did is they had 10 patients who had progressed on multiple prior lines of therapy. And this included, these were heavily pretreated patients, prior checkpoint blockade, sometimes combined immune checkpoint blockade, also prior targeted therapy. And they wiped out their inherent microbiome, gave them a fecal transplant from one of two complete responders, and saw a 30% response rate, or 3 patients out of 10 responded. And interestingly, they saw changes in the microbiome as well as in immune infiltrates. Now, this data was galvanized by a second back-to-back publication in Science. A group from Pittsburgh, Hassan Zaror and others, showed that fecal microbiota transplant could overcome resistance to anti-PD-1 therapy in melanoma patients. And so this group treated 15 patients who had progressed on prior anti-PD-1 therapy, used a fecal transplant from 1 of 7 donors, and 6 out of 15 patients showed clinical benefit to the treatment, with changes in the microbiome, highlighting some of our favorite bugs here associated with response, along with changes in serum metabolites in responders to treatment. And importantly, these approaches can actually be helpful in treating immunotherapy-related toxicity. And so Mimi Wang and others at MD Anderson have now treated several patients with fecal transplant from healthy donors to really treat refractory colitis. This is an example of one of those patients that was published in Nature Medicine, where a 50-year-old female came in with metastatic cancer who had been treated with combined immune checkpoint blockade, had rip-roaring colitis that was refractory to steroids and anti-TNF. And after treatment with a single dose of FMT from a healthy donor, she actually had complete resolution of her symptoms. I show here at diagnosis, she had a lot of inflammation, got worse on biologics, and actually got better after a single dose of FMT. Now, biopsies were done both before and after treatment with FMT and showed inflammation at both time points, but it went from a CD8 or cytolytic T cell dominant to a CD4 FOXP3 or regulatory T cell dominant infiltrate, suggesting that the treatment was associated with favorable changes in that microenvironment, which was potentially associated with the benefit. But there are issues here, as we still don't know who the optimal donors might be to enhance responses to cancer immunotherapy, and there are known as well as unknown risks with fecal transplant. And this was a safety alert regarding the use of fecal transplant for different indications. And 2 patients actually recently died after getting multi-drug resistant organisms after receiving a fecal transplant for C. diff. So based on this published data and on results from upcoming FMT trials, I think an important question is, can we actually engineer an optimal consortia of microbes that could enhance responses to immunotherapy? But published studies have actually helped to identify potential signatures that contribute to response, but there's really modest overlap. And so one of the most vexing parts of this is everyone has kind of focused in to say, okay, what's that one bug that I can put in a pill and give to patients to enhance immunotherapy response? And it's really been elusive. And why is this? Well, I think there's several different factors that can account for this. I think, different strategies for sequencing, regional differences. Certainly I always say the diet in Houston, Texas is quite different than the diet in Paris, France. But another key feature is that it's not necessarily the names of the bugs that matters, it's what they're doing. So function over phylogeny. But I think ultimately, results from additional cohorts as well as FMT and other trials will actually help to inform the optimal composition. And groups are working hard on this. So Tom Gajewski is working on a bifido-based strategy to try and modulate gut microbes. We are working with Seres Therapeutics on a strategy using a consortia, and Kenya Honda has an 11-mer that he's working with. I can tell you that these trials are underway, but they've been impacted, right? And our own trial with Seres Therapeutics was impacted profoundly during the COVID-19 pandemic, and it's actually on hold currently right now. Now, how about the role of diet? And so given the critical role of the gut microbiome, what's the role of diet and other factors in response to cancer treatment? And so this is the you are what you eat part. And so one aspect that's been shown to impact response to immunotherapy has been antibiotics. And so antibiotics have been shown to negatively impact response to immune checkpoint blockade. And Laurence Zitfigel is really one of the first people to publish this in her science paper in 2018. She showed that if patients with non-small cell lung cancer got antibiotics just before or just after treatment with immune checkpoint blockade, they actually had much worse survival. But there was a little bit of skepticism with this because people were like, well, wait a second, these patients are sicker, that's why they're getting antibiotics, and that's why they're not doing well. But now multiple other studies have been published, including this one in JAMA Oncology from 2019 from David Pinato and others, showing that treatment with prior antibiotics before initiation of immune checkpoint blockade is not only associated with worse response rates to therapy, but dramatically worse survival. And so we should definitely take caution here. And now multiple studies have actually been published really supporting this. And it's not to say that we should stop antibiotics altogether, because some of these patients do get infections and do need antibiotics, but can we use a more targeted approach? And rather than when someone comes into the ER with a fever, and potentially immune-related adverse event, rather than blasting them with broad spectrum antibiotics, can we really use a more nuanced and informed approach? And we actually have a clinical trial underway assessing the impact of perioperative antibiotics on the gut microbiota and other variables. Can a single dose of antibiotics in the context of surgery actually impact gut microbes and immune responses? And wait and see. We'll let you know. Now, in addition to this, we also studied the influence of diet and lifestyle factors on the microbiome and response to immune checkpoint blockade. And so Chris Spencer, who's a Parker data scientist now, and Lorenzo Cohen, who's the head of integrative medicine at MD Anderson, really asked the critical question, what is the impact of diet and other lifestyle factors on these components? And so in every patient that we enrolled onto our protocol, we also collected diet and lifestyle surveys on these patients. And what we found when we did this is that if patients had a high fiber diet over 20 grams a day, they actually had a higher diversity of bugs in their gut microbiome, a higher abundance of the response-associated taxa, and they were actually about 5 times more likely to respond to treatment with anti-PD-1. Now, when we layered on the type 1 signature, we actually found that diet still matters. And so it's not enough to just have a good signature. You actually have to be feeding it the right things. And we did see a statistically significant difference in patients with a high versus low fiber diet, with patients with a high fiber doing much better. So eat your fiber. And parallel studies in preclinical models recapitulated these findings. So we worked with Giorgio Trinchieri, who ran parallel studies in a preclinical mouse model and showed that if mice receive a low fiber diet, they fail to respond to treatment with immune checkpoint blockade for melanoma and other cancers, have a dramatically different microbiome. And he actually found insights into the mechanism through which this was happening, and saw that only in mice who received a high fiber diet could they activate T-cell pathways in the setting of treatment with anti-PD-1. And there is evidence that changes in diet can actually have an impact on gut microbes and associated physiology in a short time frame. This is data from Carrie Daniel, who's a nutritional epidemiologist at MD Anderson, who ran this Be Gone study, where she basically took patients, colorectal cancer survivors, and gave them 1 to 2 cups of beans per day and showed a bloom of favorable bugs in the context of treatment. However, as soon as she stopped the intervention, it reverted quickly to the pre-intervention status. Nonetheless, Carrie and Jen McQuaid and others have dietary intervention studies underway in patients being treated with immune checkpoint blockade. And testing diet as a precision intervention in cancer is a real, I think, a real opportunity here. And their hypothesis going into these studies is that a whole foods-based fiber-rich diet will actually modulate the microbiome and enhance systemic and anti-tumor immunity. And they've completed phase 1, are now in phase 2. They actually have a dietary kitchen at MD Anderson that prepares these meals for patients. They're actually incredibly good meals. But, could we actually, is there an opportunity to have a blue apron for cancer patients, if you will, to actually promote better diet in these patients? Now, another important question, though, is how do over-the-counter probiotics influence the gut microbiome and therapy response? And this was intriguing data because, an unintended consequence of groups publishing this work is that patients now want to modulate their own microbiome, right? And so everyone wants to do it. And how do they do it? Well, they run out to a pharmacy and buy an over-the-counter probiotic. And so we were curious about this. We asked our patients if they took over-the-counter probiotics regularly. We found that up to 40% of patients actually did report taking over-the-counter probiotics. And if they did, not only did they have a lower diversity of bugs within their gut microbiome, they actually had worse responses to treatment. Took this back to a mouse model and showed that if we gave mice a rockstar microbiome, a complete response from a complete responder donor, and then gave them one of 2 probiotics we bought off the shelf at CVS, it actually impaired their anti-tumor immune responses, and they actually it enhanced tumor outgrowth. And Lorenzo Cohen put out this op-ed in the Wall Street Journal where, interestingly, while we were running these studies, Lorenzo actually developed metastatic melanoma himself. He asked us to profile his microbiome, and I can tell you it was the most beautiful microbiome I've ever seen. He wanted to take matters into his own hands and started taking probiotics and had a much lower diversity and a worse profile, and actually reverted back and put out this op-ed about a year or 2 ago. And he's doing great, by the way. Now, importantly, there is crosstalk between the gut and tumor microbiome, and strategies to modulate gut and tumor microbes are currently underway. So I can tell you that in Florencia studies that she published in Cell, she found that a significant proportion of the bugs she found in the tumor are actually derived from the gut. She actually has a trial underway that's using fecal transplant from either complete responder or potentially healthy donors to change the microbiome in those patients. And multiple strategies are underway to alter gut microbes, including not only fecal transplant, but also diet or pre or postbiotics, and then microbial consortia or probiotics. But I highlight that it's complicated. And this was a publication that Jen McQuaid and I put out a few years ago, showing that there's a lot of nuances here, both with aspects prior to treatment, during therapy, how do we assess impact, and then what are the long-term outcomes here? And again, I pose, revisit the question, could the microbiome become the newest frontier in oncology and medicine with diagnostic and therapeutic strategies that target the microbiome? And I put, it's here and now, right? And so investigators, believe it or not, are developing things like smart toilets that will actually, when you sit down, send data back to your smartphone and potentially even give input. There's a 3D printed pill you can swallow to sample your microbiome, and even looking at volatile organic compounds, or these VOCs that are now using a breathalyzer for disease, if you will, and so not only from a diagnostic standpoint, from a therapeutic standpoint, can we use a personalized nutrition approach like Ron Elenov promotes, and your microbiome could even play a role in your COVID-19 response, and even in your response to COVID-19 vaccination. We're actually studying that ourselves. And we do have a program at MD Anderson with infrastructure to translate and innovate. And is the Prime TR. We are recruiting talent and also would love to collaborate with the Dana Farber Harvard contingency. So now one little final tidbit in the last 2 minutes is wanted to pose the question, how about the role of gender and sex hormones on cancer and therapy response? Are boys equal to girls? And we know that there are differences not only in cancer incidence as well as cancer mortality. And we also know that sex differences in efficacy and toxicity of systemic treatments is really kind of an undervalued issue in the era of precision oncology. And so interestingly, when we were doing our neoadjuvant studies for targeted therapy, when we were doing the analysis of the RNA-seq data from the longitudinal samples, the analyst on the phone, when we were going over the QC, pointed out to me, he said, well, Jen, did you notice that all the patients who achieved a pathologic complete response were female? And I was intrigued. I was like, no, I didn't. But we went back and looked at additional cohorts. Lo and behold, this bore out. We've now looked not only in the setting of neoadjuvant treatment, but also in patients with metastatic disease and see the same phenomenon. Took it back to a mouse model, and we find that these are hormonally mediated. And we actually have shown that if you block androgen receptor signaling, you can actually improve responses to treatment with targeted therapy, not only for males, but also for females. And in addition to this, sex-specific differences are also noted in immunity and could impact responses to immunotherapy. And this is nice data from Fabio Conforti and others. It's in Press in Clinical Cancer Research, showing that you have differences in the immune profiles of individuals, in this case, non-small cell lung cancer, based on sex. And this could really impact therapeutic response. And so I think in conclusion, we've certainly made significant progress in the treatment of melanoma and other cancers with the use of targeted therapy as well as immunotherapy, but not all patients respond. More therapeutic options are needed. I think a deep understanding of the numerous factors that contribute to carcinogenesis and to therapeutic response are needed, including factors internal and external to the host. Multidisciplinary teams, including patients, families, clinicians, basic and translational researchers, as well as foundations, funding bodies, and pharma are all key in advancing the field, and we need to work together to optimally drive this. And finally, there's still a tremendous amount to learn, but the future is bright. And so with that, I'd like to first thank everyone for participating and joining. Thanks, Sandy, and the organizers for the opportunity to present at the Connect Science seminar here. Thank the army of people who really contributed to this work, and also wanted to leave some time for questions. So also wanted to thank my awesome family, who's silver lining of the pandemic. I'm not traveling anymore, so they're happy to have me home. I'm even coaching softball right now, so it's pretty awesome. So some improvement in quality of life. So, but with that, I'll take any questions.
Wow, thank you. That was so great. I wish you could hear the applause, probably the standing ovation that you're getting right now. That was phenomenal. Thank you. I'll start right in with questions. And yeah, thanks for leaving time. We can get to a number of these. So Peter Engel is asking, does the gut microbiome change in response to anti-tumor therapy?
Yes, the short answer is yes. Now, in our anti-PD-1 cohort, we didn't see a lot of shifts in the microbiome, the gut microbiome over time. So it depends on the therapy. The short answer is yes. The long answer is it depends. Now, interestingly, there are changes, even when in the preclinical models, you can even see a change in the gut microbiome when you implant a tumor. So there's this crosstalk, Now, in the setting of treatment with combined immune checkpoint blockade, you definitely see more changes. And in the setting of treatment with chemo and other strategies, you definitely see changes. But it's very treatment dependent. And there are other variables that go into it too. But, short answer, yes. Long answer, it depends on a number of factors.
Great. Yeah. So that prompts me to ask my own question about what are the other sort of sledgehammers that you know about to the gut microbiome, diet, sex hormones, chemo. I have to ask about age.
Yeah.
If you've seen age stratification, but what are maybe some of the other heavy hitters that impact the microbiome that you know about?
Yeah, exactly. And I think now this opens up everything, right? Because you have to think about everything. And it's like, I've changed how I eat and how I feed my kids. And it's like, everything we put into our bodies actually can impact our gut microbes and, our risk for cancer, and other aspects and how we metabolize things. So I think, it changes everything. So what can impact it? Well, I think certainly diet and other factors, antibiotics, you wonder, like antibiotics in food or other aspects, could that even impact it? And I think that's a real potential. Medications, and we're looking at what medications. Stress, can impact the gut microbiome too. So, and we and others are kind of taking that into consideration. incorporating profiling of the microbiome, of diet, of these other lifestyle factors. Ultimately, can we actually use, technology to advance this? Can we, have transcutaneous sensing of metabolites that are produced by gut microbes so that on my smartwatch, it'll measure my metabolites, send me an alert, be like, all right, Jen, eat a little more kale today, hold off the red wine or something, can kind of give us real time, information about, and intervene, and help us, enable us through technology. And I think that's really where there's a lot of hope, and I think tremendous hope for overall precision health, far even beyond cancer.
That's really cool thought. I can see it happening. And I think my watch right now would be screaming at me because I just ate a handful of peanut butter pretzels as my lunch. So, but let's get on. Zebronai has a question or a comment. Great talk. Scoop is in the poop, but will actually be in the metabolites produced by the bacteria in the poop. Having identified bacterial signatures, how far are we from defining the metabolites that one may use to skip the poop for the real scoop?
Yeah, exactly. No, Zev brings up a great point. And it's like, FMT, fecal transplant, even consortia, I think consortia have some hope, but I think, that's not the end game, right? Like this is a stepping stone, right? So how can we get to that next level, to the metabolites, and can we even take someone's inherent microbiome, ex vivo engineer it, similar to CAR T therapy, right? Can you actually engineer someone's gut microbiome, put it back in, and really have it performing the right function? Because again, it's not the names of these bugs that matters, it's what they're doing. And so can we alter these microbes to enhance their function, or can we come up with next gen therapeutics that are, postbiotics or metabolites? And, Zeb's doing beautiful work on it himself and on prebiotics as well. So, I think, the answer is to work together to accelerate it and learn as much as we can and then test it together.
Yep. Excellent. And I have to apologize if I'm not pronouncing your names properly. I'm asking you questions. But we have another question from Oluwakemi Taiwo. Does blood type play any role in shaping the microbiome?
That's a great question. We have the cohorts that we could potentially look at it. We ourselves have not looked at it, but we would love to look at things like this with other investigators. There's only so many hours in a day, and and so many hats that we all wear. It's like, I think the key to these studies is to collaborate, to get these published, to deposit it so that other people can mine this data and learn, because, we owe it to our patients to share this data, to learn together, to work together as a global team, to really advance cancer care and overall precision health.
Well put. Thank you. Saman Maleki asks, do you think would sex be a factor in better microbiome engraftment between stool donor and recipient?
That's a great question. And I know Saman's doing some work in the space, interestingly, another fun tidbit, so you get more FMT donation out of a male than you do a female, so interesting another cocktail party tidbit for what it's worth. But yeah, no, I think that's a great question. We ourselves have not looked at, from a recipient, donor versus recipient standpoint, is it male or female better? What's the impact of hormones? I'm not sure. I think it needs to be tested, but great point.
Yeah. Nicolas Prieto, what about increasing the amount of fermented foods in the diet with natural probiotics, yogurt, kimchi, et cetera, to increase microbiome diversity?
Exactly. And I think that's potentially useful. I think it needs to be studied. It's, again, like with the probiotics, are all probiotics bad? Not necessarily, not all over-the-counter probiotics, but if you think about why they were formulated early on, it's because they were easy to grow and put into pills. It's not necessarily because they were known to stimulate an immune response, or increase mental awareness or what have you. So but yeah, I think more science is needed behind it. So we need to not only track what our patients are taking, not only medications, but also diet, supplements, et cetera, enable technology to help us to do that, and then to learn from it and do interventional studies, the right interventional studies, and use preclinical models. I think these are incredibly useful.
Mm-hmm. Thanks. Candice Lay is asking, what an amazing presentation. How does the diversity of the gut microbiome specifically affect the efficacy of immunotherapy in the setting of pancreatic cancers, given the stromal immune interaction in the tumor microenvironment can influence the efficacy, but is largely unknown?
Gotcha. Yeah, and it's curious, right? It's a hard question. So from a gut microbiome standpoint, diversity matters, right? Diversity matters in all of life. But it's, if you think about the gut microbiome, why is a more diverse gut microbiome better? And if the way I think about it is you have more functional redundancy with regard, to the gut microbes. So if you have a hole in your gut microbiome repertoire, you can fill it more easily if you have a more diverse microbiome. But why would a more diverse tumor microbiome matter? And Florencia actually showed that if you transplant in a fecal transplant from a long-term responder into a mouse model of pancreatic cancer, the tumors grow slowly and they have more immune infiltrate. Now, there could be some element, of molecular mimicry. Is there some interaction? And, as we'd seen with Yardenia's work, there's, microbial neoantigens that are actually being processed and presented. Could that be contributing? Potentially, but I think there's still a ton to learn.
Yeah. So, one final question that I promised some of the trainees I would ask, because we had a really interesting journal club yesterday, a paper that identified gene expression signatures in response to therapy. Asking really, what do you envision, or what do you think it will take before all these, biomarker identifications or gene signatures that predict response to therapy, or, signatures of the microbiome, et cetera. How do you envision that will actually become clinically utilized and meaningful?
Right.
When
that's
can we start using these signatures?
Yeah, exactly. Because, PDL1, tumor mutational load, you have all these different biomarkers of potentially, microbiome response to therapy. You have to take it in totality, right? And so, how can we come in with an integrated approach to look at not only host immunity inherently, in your genetic composition, along with the exposome, like what have you been exposed to over life? How does that impact how you either develop or cancer or respond to cancer treatment, or what's your risk for cancer based on that? And then what are the modifiable factors that you could change today that could impact that, diet, other factors? I think it starts with doing the profiling and taking data sets and integrating those and then also using models to be able to say, okay, what mix of biomarkers and how can I assess those, in an easy and cost-effective way. That's always the hard part, right? Is ultimately getting there. But can we, work with, Harvard, MIT, other great groups around the world to develop, different biosensors or, different technology that could actually really monitor this? I think, how do you inform that? I think by looking at these published and prospective cohorts is helpful, and ingesting that data and then using that to inform the development of these approaches, and then testing it in models along with in patients ultimately.
Well, thank you. I threw sort of a tougher one at you, but
yeah,
it's really interested, in asking that question. And
it's not a one size fits all. I think, sure, microbiome's a great biomarker in theory, but we don't know that, it needs to be tested, and it's not a standalone one, because it's like you could have a great microbiome, but if you have, a tumor profile from a genomic standpoint, that's, you need to be able to know that too, to integrate that in.
Absolutely. Yeah. Well, thank you again so much, Dr. Warga. On behalf of everyone here, I can thank you for your time. And, we were really thrilled that you were able to come here today and present, or be on screen today.
Yeah, I know. I wish I could be there in person.
Yeah, so do we. But thank you again.
Okay. Thanks all. Be well.
See you.
Take care.